Hello everyone. Now we are going to discuss about the fertilization in angiosperms. Fertilization is a process in which by the formation of the male and female gametes, by the fusion of the male and female gametes, the zygote will be formed here. That zygote will be developing into the embryo and it will give rise to the fruits, flowers, seeds, all these things here. That is the fertilization we are saying. In the angiosperms particularly, how the fertilization takes place. What steps it has been involved, we are discussing now. Before going into the fertilization of the angiosperms particularly, first we have to know about the embryo sac. This is the structure of the embryo sac here. Embryo sac is the female gametophyte in the particularly angiosperms, in the plants here, right? If you see the embryo sac now, embryo sac is always formed by the megasporogenesis phenomena. Megasporogenesis gives rise to megasporangium. That megasporangium we are calling as a embryo sac. Embryo sac is always 7-celled, 8 nucleated. It is consisting of 7 cells and 8 nucleus. Now we will see where 7 cells are there. We will see here 1, 2, 3. 2 synergids, right? 1 XL, 2 synergids, 1 XL, 3, and 1 nucleus, 4, 3 antipodals. Totally 7 cells we had. 7 cells, 8 nucleus. It means what? Here in the polar nuclei, 2 nucleus we had. That's why we are calling as a 7-celled, 8 nucleated. Very important point here. Embryo sac is always 7-celled and then 8 nucleated. Here, if you see the embryo sac, the side towards the antipodals we are calling as a chelazal end. The side towards the synergids and XL we are calling as a micropylar end here. Then synergids are the 2 cells which are present in the embryo sac. Synergids are consisting of what? Filiform apparatus. This filiform apparatus will guide the way for the pollen tube to enter into the embryo sac. Then XL, right? It is participated in the fertilization here. Polar nuclei consisting of 2 nuclei and antipodals and the chelazal end we have. This is the structure of the embryo sac we are saying. Now we will see the 3 steps how the pollen grain will reach the particular XL or else polar nuclei, right? Here by the 3 methods, mainly the pollen tube is reaching or else pollen grains are reaching through the pollen tube here. 3 methods: chelazogamy we had, porogamy we had, mesogamy we had. As we know, chelazal end we had, we know, micropylar end we know. First of all, if you are going for the porogamy here, porogamy it means what? If the pollen tube is entering through the micropylar end like this, right? Pollen tube is entering into the embryo sac through the micropylar end, we are calling as what? Porogamy we are calling it. Right? Like this it is entering and it is fusing with the XL and then polar nuclei. If the pollen tube is entering through the integuments, these are the integuments here. These are the integuments. If they are entering through the integuments, 2 integuments, entering through the integuments we are calling as what? Mesogamy. Right? Pollen tube is entering through the integuments we are calling as what? Mesogamy. If the pollen tube is entering through the chelazal end like this, chelazal end we are calling as what? Chelazogamy. By these 3 methods, what is happening? The pollen tube is entering into the embryo sac. The pollen tube is consisting of 2 pollen grains. 2 are in a haploid condition. Remember this point here. And then, now this is about the introduction here. Now we have to discuss about the 2 steps again. Syngamy, triple fusion, and double fertilization. Syngamy, triple fusion, double fertilization. These 3 are the important steps in the fertilization in angiosperms here. First of all, what is the syngamy? Syngamy is a phenomenon in which what is happening actually, as we know, pollen tube is entering by the 3 ways we are saying, right? Chelazogamy, porogamy, mesogamy. Through the pollen tube is consisting of how many pollen grains? Pollen grains are the male gametes here. 2 pollen grains. 2 are the haploid here. In that thing, 1 haploid pollen grain, haploid male gamete, haploid pollen grain fuses with the XL. Haploid pollen grain fuses with the XL and gives rise to forms a zygote here. This phenomena, this condition, this process we are calling as a syngamy, right? After syngamy, what we are saying? Triple fusion we are saying. Triple fusion, it means what actually? Here what is happening? The pollen grain which is there, 1 pollen grain will fuse with the XL. Here, remember the point here. 1 pollen grain is fusing with the XL, other pollen grain is fusing with the polar nuclei. Right? Here, what is happening here? One pollen grain is fusing with the XL, other pollen grain is fusing with the polar nuclei. When the pollen grain is fusing with the polar nuclei, when the pollen grain is a haploid gamete, that means haploid. These are the two haploid nuclei again. Three haploid nucleus, they are involved in the fusion of the polar nuclei condition. Hence we are calling as what? Triple fusion. Three haploid nucleus are involving, right? That's why three haploid gametes are involving in the fusion. Hence we are calling as what? Triple fusion. Double fertilization, it means what? Double fertilization is a phenomenon in which what is happening? Double fertilization, it means two times the male gamete is fusing. One time with the egg cell, other time with the polar nuclei. As two times the male gamete is fusing, as two haploid male gametes are there, we are calling as what? We are calling it as a double fertilization. This is the phenomena which takes place in the fertilization of the angiosperms, right? It is a different concept here. In this fertilization angiosperms, first we have to explain about the structure of the embryo sac, how it is formed, and about the chelazogamy, porogamy and mesogamy, and three steps mainly. Syngamy. What is syngamy? The haploid pollen grain which is fusing with the egg cell leads to the formation of the zygote. That phenomena we are calling as what? Syngamy we are calling particularly. And triple fusion, it means what actually? Where three haploid gametes are fused. That phenomena we are calling as a triple fusion. Three, right? Two polar nuclei, two are in haploid condition, one pollen grain, three haploid gametes are fused here, hence triple fusion. Double fertilization, it means what? One pollen grain is fusing with the polar nuclei, other with the egg cell, secondary nucleus or polar nuclei. As two times the male gamete pollen grain is fusing, hence we are calling as a double fertilization phenomena. This is about the fertilization of the angiosperms. Very important concept, a very important topic for the 8 marks here.